That's going to be the one that's involving more critical thinking. These first two chapters are very straightforward. That will get the critical thinking part, which is why I want to get through a lot of this week, okay? Because next week is it. So on Monday, we'll finish that, but we'll be doing, when we look at chapter 26, which is sort of a sex chapter. I don't even know the name of the chapter. I just call it sex. But it's not much sex. It's literally going to be, we're going to just be doing endocrine control of sex hormones. So it's really a continuation of that same way of thinking. Usually that lecture itself is like 20 minutes or something. So overall, we'll be able to finish and have plenty of time. I put in, again, the extra questions, which I've updated, is, again, everyone should have got my email, because I decided to add, again, some of those multiple choice. Some of those are literally on the test or something similar to get that idea of the complexity of it. Most of those you'll see, hopefully by this weekend you'll see, are going to be on chapter 23. Okay, so you can practice doing that. So we will have time again, because we have no lab to review, for the exam potentially, that day as well. Again, I'll stay as late as, just like last time, I'll stay as late as need be. But you can't, obviously, you can't get behind here. I mean, there is no more time to get behind. So we'll do all that. As far as before class and even after when people go leaving, I have all the tests. If you're doing the cumulative, you can look at your old test and get an idea, talk to them about it. You also can look sometime this week at your exam 4. I had something like, I have to do the percentage, something like 65% got A's or 70%, some amazing amount got A's, and almost everybody else got B's, so you guys just really kicked some butt, okay? You really got this stuff, higher than any other class. Sort of what you reached that challenge and sort of, surpassed it. So it was really, really good. And anyway, I was happy about that. Hopefully that made your holiday a little bit better than worse. So let's do this. First, again, first two chapters, I can't jump up and down. It's not the kidney. So just pay attention to what I want you to know. That's going to be sort of the key here. And we'll get through a lot of this today. Also, at some point near the end, you get to do my reviews, student reviews. And the only one I really have to do it, let's say, because it is Monday lab, because in case we go over, is to have my Monday group do it. But if you want to do it, you're free to stay and actually review. Get payback is what they say. So so you do get to review me and the class today. Okay. So let's do this. So digestion. This is actually pretty appropriate for the holiday. I should have put a turkey on. What was I thinking? This is my old slide. Okay. But we're going to talk about digestion. This first slide is for sort of just overall informative. Again, you're not memorizing this, but we're going to actually take us through the digestive system today and what basically happens. But what I find sort of interesting about this is that if you look at all what is going to be secreted into the GI tract and look at sort of the fluid levels of it, it's about 9 liters. Okay, remember, we only have 3 liters of plasma. That's a lot of fluid. So clearly, we have to absorb most of it back. And that's why most of it will be actually, the workhorse of all this will be the small intestine. And again, it's sort of, it's one of those numbers like 180 liters, numbers which seem really, really high, that you wouldn't necessarily think. It also reminds me that, when you think about, let's say, severe diarrhea, especially, let's say, in a small child, why it's just deadly, okay? I mean, you're losing a lot more volume than just being dehydrated, and you're going to become quickly dehydrated, from this process. But that's that. Just like the kidney has sort of these processes that we're going to go through, we're going to talk about digestion, and I'm going to define it in a minute. There's motility, which we're only going to have a couple slides on, so I'm not going to really emphasize motility. But we're going to be doing kind of the same thing. We're going to talk about things which are secreted into the lumen of the GI tract, just like we were talking about the kidney. And now we're not going to call it reabsorption, we're going to call it absorption. And this is going to get it from the lumen of the GI tract into the blood. So it's going to be very similar. So the only 2 slides on motility. Okay? Again, if you're reading the book or even some of the notes, there's more material than I want, are just these 2 slides. So for motility is the idea of, and we'll show this picture here in a second. that smooth muscles. So we know muscles, it's going to be the smooth muscles. We already know sort of this idea that smooth muscle has sort of the sympathetic, parasympathetic, and sort of local hormonal controls, right? This is sort of all news to us. That's fine. The other thing is that some of these cells within the GI tract actually, kind of go back up to this, can spontaneously reach X potentials even without neurohormonal signal, just like it did in the heart. And there was a question back on that cardiac test that everyone loves. Okay, forget about that cardiac test. Okay, but there was a question in which you're supposed to sort of compare pacemaker potential to slow wave. And the whole idea of slow wave, which is what we're talking about now, is that they don't, this is spontaneous, but it doesn't always reach X potential. That's the difference. It's the same thing here. So these slow wave potentials are not in every cell. They're only in specific cells in the GI tract called the interstitial cells of Gajal. And when they reach X potential, they're going to spread to these other smooth muscles through gap junctions. And then we're going to have this peristolic movement. But that's all we're talking about. Okay, so we're not going to go into the different, peristalsis or anything else. But these are the only two things for that, when we're looking at these concepts for motility, there's really just this one slide with that figure. So, digestion, which is next. By definition, and we'll put it up here, digestion is going to be the breakdown of food into absorbable units. Something that we can actually absorb. So the first thing we're going to summarize on here are what the absorbable units are for the three main food groups we're going to do. And so we have carbs first. So I sort of put the bun here, put the stuffing. I don't care what you put. But we have carbohydrates. So the carbs. Again, for the carbs, we're talking sort of polysaccharides. This is reviewing stuff we did sort of at the beginning too. But the absorbable units will be monosaccharides, singles. With, again, sort of glycogen and starch being sort of the typical polysaccharides we're going to eat, and we're going to digest them into monosaccharides. Again, with glucose being sort of the central one. Now, you're not going to have to, when we look at this, you can see there's, we talk about that there's 3 disaccharides in our diet, and, they're made up of these monosaccharides. We're not memorizing that at all, okay? But we do want to start, for each one of these, look at sort of the enzyme which breaks it down, okay? And so one of the main enzymes here that you do want to associate with this is amylase, which is actually in saliva. So we're gonna begin digesting, begin breaking down carbohydrates right away with amylase in our saliva, and eventually we're going to get to monosaccharides. These are the absorbable units for sugars. And that's all you need to know from that slide. Okay, the protein. So this was carbs. Here's protein. The absorbable units are amino acids. And in parentheses, we'll put small peptides. So, and even with this slide, and when it comes down to it, a protein is hundreds of amino acids long. We have to start cutting them with these different enzymes. I actually don't want us to know these names. I just want us to know the general name of the enzymes breaking them down, which are called proteases, sometimes called peptidases, And later when we go through the GI tract, we're going to put some specific ones up here. But you don't have to know endopeptidase or aminopeptidase. Those are just specific types. But I do want us to know when we're breaking down proteins to its absorbable units, amino acids, we're going to be using various proteases or peptidases. And that's all we need from that slide. These two chapters are very board-oriented as far as what you need to know. You'll see that. So I'm trying my best to kind of write it where you can almost read it.
What's the polymer for protein, the actual protein?
The protein is just the polymer, yeah. So lipids. We're going to put it up here as lipids, which are fats. Again, it's the grease of the double-double, so to speak. Remember, our major energy storage, so again, we're reviewing stuff from sort of the beginning, are these triglycerides, which have a glycerol backbone and 3 of these long fatty acids. And so for lipids, we're really looking at those, the triglycerides. And using, that's why I put lipids up here, enzymes called lipases and its friends, co-lipases, but lipases is fine. The absorbable units for lipids would be sort of free fatty acids, and sort of having the glycerol with 1 fatty acid or a monoglyceride. And that's the first major thing that we did in this chapter, okay? Is basically, what are the absorbable units for carbs, proteins, and lipids, and what are sort of just the overall name of the enzymes that digest them?
So for lipids, the enzymes, the free fatty acids?
The enzymes are what breaks them down. The enzymes are the lipases, okay? The absorbable units are the free fatty acids and the monoglycerol. Okay, what we have left in the chapter is sort of taking us through. We're gonna start what happens in each of these places. You know, I put this up here. We're not memorizing this because there's plenty of stuff here that I'm just simply not going to talk about. But it is sort of a nice chart because it has all of the processes, motility, secretion, etc., and it says what happens in each one, and that's sort of nice. But we're only gonna cover a couple of these. So you look at my slides, on these. You can see the bulk of the work for the next 20 minutes or so. It'll only take about a half hour for this lecture. Will be in the small intestine. That's where most of the digestion, most of the absorption happens, and that has most of the stuff. So we're just following the double-double. And literally, this is sort of the slide for this one. So remember, just to go back a second, we're really not talking about motility here, but that's what, obviously that's happening. But when we come down to it, the secretions are saliva. Okay, saliva contains amylase. So we're going to begin digesting carbs and some lipases. So we're going to begin digesting some fat right away. That's what the saliva, gets the process going. The absorption, here it says none. It's really basically minimal. So some glucose, maybe some drugs can get absorbed. I'm not going to try to fool you on this, okay? I mean, it's just that, it was a little off on that. But that's it. So that's all you need to know about what happens in the mouth and esophagus. Amylase containing saliva containing these 2 enzymes, and therefore we're going to begin, digesting those. That's it. When it comes down to the stomach really, as we're going to see, doesn't digest and absorb much food. It preps it and stores it. We're going to see, food is going to be digested in and absorbed in the small intestine. We don't want all that food just to fly into the small intestine. We won't be efficient. So that a lot of the function of the stomach will be kind of prep things, but store it. So that almost like a toothpaste, we can start to squeeze it out into the small intestine bit by bit. So what you need to know for the stomach is basically what's up here. It's going to be these 3 cell types. there's other cell types if you look at the chart. These three cell types and what they secrete, what they're putting in. And, it's pretty much up here, but I can sort of summarize too. If you look at chief cells, okay, a main thing that they're secreting is something called pepsinogen. Is pepsinogen active?
Yes.
No. Okay? It is a zymogen. It needs to get converted to pepsin to be active. And, speaking of that, the parietal cells are going to secrete hydrochloric acid. I'm going to talk about that in a second. But I want to put it up here because that's what activates pepsin. Okay, as one of its functions for the HCl, and I'll talk about some of these others. So these two cell types right now are working together. The chief cells are producing pepsinogen, which is inactive, but the hydrochloric acid from these cells are going to activate it. Pepsin is a protease. So it's going to start digesting proteins. So I've even kind of put this here overall too, kind of examples of this. So pepsin is a specific protease, and so remember right now that it's in the stomach. Okay, hydrochloric acid. A couple things about this. The one thing is, kind of put this over here in a sense, that the stomach pH, because of all this hydrochloric acid, is pH 2. What is the pH in normal blood fluids? 7. So pH 7, we'll put blood. Again, this is the stomach gastric juices. So, which one has more hydrogen? How much more? 10 to the 5th. Remember, if you look at the scale, 6, 5, each number is a factor of 10. So it's 10 to the 5th, so that means we are concentrating the acid in our stomach 100,000-fold. Does that take energy? Yes. And if you look at the next slide, which you're not going to have to memorize, but we want to see our friend one more time, although it's hard to recognize there. How we're going to do this is the following. Where is this thing? CO2 plus water. It's going to produce what? Hydrogen and bicarbonate with carbonic anhydrase. Does that seem like something we've seen before? Again and again and again. Okay? So it's also in these parietal cells, and so you're not going to have to redraw all this or really know it. I just wanted to show you it. So we're going to use that same type of reaction. This time we're going to use a ton of energy, ATP, to pump hydrogen in 100,000-fold. Chloride is going to come in because of the electrical gradient that's created, and that's how we're getting all that hydrochloric acid. Now, you don't have to redraw all that. We're not going to do it anymore. I just wanted to show you, again, how important that one enzyme and that one reaction is. We've seen it 5 times in the last 2 months. Okay, so the other functions here are listed for this hydrochloric acid. It's going to help kill bacteria. We're open to the external environment, so it's nice to have a sort of a pH which will be not conducive to growth. It also denatures proteins. And the idea of that is that you have this 3-dimensional protein structure. It's hard for these enzymes to get in. By the hydrochloric acid, will literally linearize it, and that's denature. So by changing the pH to pH 2, we're going to denature the protein, and then all the proteases can start clipping along and make it easier. So, chief cells, pepsinogen to pepsin. Parietal cells, mainly hydrochloric acid to perform those functions. But we need to protect the the cells of the stomach themselves from the pH2. And so it's mucus cells. And we're going to show a picture. They secrete these two things, mucus and bicarbonate. And here's sort of that idea. The gastric juices being pH2, we don't want, unless we want an ulcer, we don't want our cells to actually experience that. So those the cells will then produce, again, whereas the mucus has a physical barrier, and then again, we're going to see this a couple times, bicarbonate, again, is a buffer. It will neutralize the acid so that at the cell surface there, it's pH 7 under normal conditions, and our stomach is happy. Okay? If we have sort of ulcers there, some of this gastric juice can start degrading our stomach lining, which would be a bad thing. Okay? Now, that's the main thing for the stomach.
Sorry if you already said this, but what exactly does pepsin do?
It's a protease that begins digesting
right.
proteins.
Okay.
I just kind of put them over here. And speaking of that, overall, sort of here's the end of the stomach. That some digestion occurs, mainly being protein and some fat, and very minimal absorption. So really, again, for the stomach, we're talking the secretions of those three cell types, the mucus, parietal, and chi. That's sort of what you have to know or remember.
Is this where most of the secretion occurs?
No. That's about to happen. Okay, so most of everything is going to occur in the small intestine. And I'm going to redraw a picture for this. This is where, again, we're almost done with this chapter, believe it or not. This is where most of the digestions, most of the secretions, most of the absorptions are going to happen in this small intestine. And that's why we have it very long, surface area, convoluted, etc. Because this is what we want to, this is where the site of action is. I'm going to erase actually this because it's pretty easy. Because I'm going to sort of draw a picture, because what we're going to see here is not only are we going to talk secretions from the small intestinal cells themselves, we're going to get help from the pancreas and the liver. And so that's why this system here is a lot more complex. This is the bulk of things. There's going to be another picture which will help this in a minute too. But overall, here's the stomach. And it's going to be sort of releasing this pH2 partially digested food, which sort of is called chyme. It's going to basically put this into the small intestine. But we're going to have the pancreas secrete some things. Actually using ducts. That's with T, not quack, D-U-C-T-S. There's literally ducts. We'll kind of show a picture that are going to secrete things. And we also have the liver making bile, which we'll see, which will be stored in the gallbladder. And we're going to kind of do this. And again, using ducts. It's going to secrete something. So we're going to have all three of these secrete things, but they're going to work together. Now, I'm going to use this picture for a reason in a minute. But first of all, I'll do this. So here are the small intestinal cells themselves. The small intestinal cell are going to secrete bicarbonate.
That thick line is the cell?
Yeah. So these are the cells actually secreting something into the lumen. It's going to secrete bicarbonate to neutralize the acid. Okay? So, and again, I put this up, but I have sort of the words too, overall too. Bicarbonate. It's going to neutralize the acid. Okay? The other thing it's going to make, and that's why, let me just put this picture here. The other thing the small intestine is going to make is an enzyme that we're going to see in a minute what it does, called enteropeptidase. which again, we're actually going to put what it does, but there's the spelling of it if you can't see my writing here, if you don't have a handout, enteropeptidase. So the small intestinal cells are going to secrete two things: bicarbonate to neutralize the acid, and enteropeptidase, which I'm going to show you what it does in a minute. Okay? Well, I guess we'll do this now. The pancreas is going to release two types of things. It's going to release more bicarbonate. Okay, so this is the pancreas secretions. It's going to release more bicarbonate to neutralize the acid. So we see we have two sources of that, right? The small intestinal cells themselves make it. The pancreas secretes it as well. We want to neutralize pH 2 and make it pH 7 for the small intestine. The other thing it releases is basically what I'm going to call trypsinogen and friends. It's going to be basically a mass of zymogens for digestion. Now we don't want the pancreas to dissolve itself. So the key is we're going to secrete zymogens with sort of the leader here being something called trypsinogen as inactive form. And once this happens, once it enters here, it's the enteropeptidase that will activate, convert trypsinogen to trypsin, which is active, and that's going to activate then all these other ones so that we can digest. It should make sense. We don't want any of this happening until we basically have food enter here. We want it all right here. So it's a combination. We're going to secrete all these enzymes, but as zymogens. Then enteropeptidase, once it's made, will convert it to trypsin, and that will activate everything else. And you're not going to have to memorize these, okay, because they're just more enzymes. Trypsinogen is the key one there. And so once it's converted to trypsin, that will activate the others. So that's why the one you want to remember here sort of is trypsinogen.
Why do you put 2?
Because that was the second secretion from the pancreas.
Oh.
The secretion, at least, to try to do it. Secretion number 1 was the bicarbonate. Secretion number 2 is sort of trypsinogen and friends, but we're going to put trypsinogen. So remember, trypsinogen is inactive, and enteropeptidase will convert it to trypsin, and trypsin is a protease. That's going to clip and activate all these other zymogens so we can digest fats and proteins. Remember, we just don't have to know the names of these. There's no reason to know those extra names. But you have to know this overall process. But while we're here, let's go ahead and put, so we can sort of remember, that pepsin was a protease that was in the stomach. We want to remember trypsin is a protease that's in the small intestine.
So this is the beginning of the small intestine.
Yes. It's actually the very first segment, which is duodenum, and that's where most everything's actually happens. Most of the digestion is actually happening right here. Okay? Let me review that too, because we don't really have much more in this chapter. But this is sort of the hardest part. And then we'll talk about bile. We'll talk about what that does. So you think about this. We just did the stomach. We started to digest, but got really things ready. We have pH 2 chyme. So we have the pancreas is releasing bicarbonate to neutralize the acid, and trypsinogen and other zymogens to get things, that once they're activated, things are going to go. But here they're inactive. The small intestine itself will secrete bicarbonate to neutralize the acid, but then it secretes enteropeptidase, which will convert trypsinogen to trypsin, protease, and that activates all the other enzymes. Chop, 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 digest, digest, digest. Okay, like a cascade. Okay. What does bile do? So gallbladder, in fact I probably put a picture up here. The other secretion from the gallbladder, the liver makes it, gallbladder will store it and secrete it, will be bile. And we'll kind of show a picture of this. What bile will do is something called emulsify fats. Let's see a better picture of that. But the bottom line is, if you look at fat, let's say it was your double-double, fat doesn't like water, and so you've got this huge fat droplet. And your lipase is saying it's hard to get in there. By using bile, it literally will break those up into sort of smaller fat droplets. That's sort of what emulsify means. We can't dissolve fat, but we'll emulsify it. We'll break it up into smaller ones, and then the lipases can do its thing. That's why bile is really important for your ability to digest and absorb fat. If you have gallstones or your gallbladder removed, it's much more difficult to eat fat. Okay, you're going to get sick, basically, because you just simply can't digest it very well. And if you can't digest it very well, it's going to continue on, take some water with it, you will be uncomfortable. Okay? Any questions on that? That's the hardest part of today, actually, is basically understanding this. Sort of the three major secretions that are happening here in the small intestine. What the small intestinal cells make themselves, what the pancreas secrete, and then what the liver secretes. Chyme, it's just what you call the partially digested food coming from the stomach. They actually call it chyme.
So, and for this purposes, are you just like saying the liver and gallbladder not as one, but just like that the bile is the main thing?
Yeah.
So is it bile going from the gallbladder to the liver? Or
Yes, via the duct, whatever duct it is. Yeah.
So, because I remember learning that with those two things and how they work, but then the liver actually secretes it into the stomach.
No, the liver secretes. The liver makes it, stores it in the gallbladder.
Oh.
It's the gallbladder that will secrete it, from that storage into the small intestine. Most of the digestions, most of the absorptions happen here. Okay. So of course this is only for illustrative purposes. If the purpose of the system is to absorb into the blood, what do we need? Maximum surface area, lots of blood vessels, right? Just structure, function, revisit again. That's why you have these microvilli. These are not cilia. These are microvilli with all kinds of cytoskeletal elements. But that's sort of, again, maximize absorption, okay? Maximum surface area, direct access to the circulatory system. So that was just like, okay. So how do we absorb carbohydrates? Do we use membrane transporters? Do those look familiar? Okay? We use, what is that guy there?
Salt.
Sodium potassium pump. Here's some transporters. Do we really want to learn that again?
No.
No, we don't. So we don't have to. But that's what we're using. We're using membrane transporters to get the sugar in. And it goes into the blood. How about the amino acids? Yeah, about the same thing. Okay, so do we need to know that again?
No.
Okay, all these are words. Here's the figure that we need to know. And let me just turn the lights off, because really it's listed well on here. I don't have to rewrite it. Both these last tests are multiple choice, so it's not an essay question here. It's going to be probably list these in order, 1, 5, 2, 4, 3, whatever it is. But let's look at what happens, because we're going to talk about sort of the combo of how we're digesting and absorbing fats. That's the hard one here. The other ones, again, membrane transporters, we've already done. So all of this here, but let's kind of follow it. Here's the, again, idea that fat will basically be not associating very well with the fluid, because again, it's a lipid. And so we have these large fat droplets. There's sort of the picture of these bile salts emulsifying the fat, so that now the enzymes can actually digest them into monoglycerides and free fatty acids, wherever that is.
So what is this called? The steps of digestion?
And absorption. So it's sort of, because it sort of combines the both. This is sort of the digestion part, like 1 and 2, I guess, up here, right? But basically, it's emulsification and lipases digest. And that's sort of the picture of seeing this, what emulsifying means. So it associates, they're smaller and these enzymes can work. Okay, here is sort of the interesting process, okay, of how we're going to absorb it. And, it's listed here, it's very well listed, but we can kind of follow it. monoglycerides and free fatty acids are lipids. They can diffuse into the cell, okay, which is 3A. Now, we're going to use cholesterol, and cholesterol actually needs a transporter, even though it's a lipid. Okay, so that's 3B. Cholesterol is transported. Now, here's sort of the interesting thing. We're going to combine it, all these things together, plus protein, to make a complex particle called a chylomicron. So chylomicron now has both triglyceride, cholesterol, and now protein, and we have this particle now that has all of that, and that's called a chylomicron. And then the last step is different too. Instead of going into the bloodstream, which is what the amino acids and glucose do, we don't want these big chylomicron particles entering our blood. That seems, bad. So it's going to go into the lymphatic system and then slowly kind of enter the bloodstream from that. So we don't want all this fat from that double-double just blopping into our blood, okay? These big chylomicrons, okay, that would be the end of it, okay? Even for you young people, maybe. And that's the process. So this is one that's a little different. And again, after that, it's going to go to the liver and do a bunch of things. It's cool, but we're not going beyond that. Here we're going simply through the digestive tract. What happens? So for fats, we're going to emulsify it with bile. We're going to digest it with the lipases. And now this complex absorption here, where we have the free fatty acids and glycerol, monoglyceride, diffuse. We'll use a transporter to get cholesterol in. We'll combine all this with protein to form a chylomicron, and that will be absorbed now into the lymphatic system. The main reason is that, if these triglycerides and cholesterol too are floating in the blood, they're not going to interact with fluid. But proteins will make them a little more soluble, more manageable to be carried. Eventually, it's HDLs and LDLs and all kinds of stuff that you can deal with later, but we're not going to deal with that now. So this is just getting into the blood. Now what happens after that? Any questions about that? So again, absorption of proteins and carbohydrates, not important. I mean, you already know it. Remember transport. Here's the one you need to know that might be different. All multiple choice questions though. What happens in the small intestine? Oh, well, first of all, most of the water is going to be reabsorbed in the small intestine back into the body. How are we going to move water? How can we move water from the small intestine back into the body? How? Yes. We're going to pump salt in. Water is going to flow by osmosis. Okay? Do we need to know all those? God, no. But that's what we're going to do. So we're going to use energy to do it. Our friend, wasn't I hot on sodium potassium pump from the beginning too? I said it's in every cell doing something different. Yes, it's in every cell doing something different. So we're going to use that for energy, and all these other ions are going to basically follow it, and then water follows where salt goes. So don't need to know that. You all do know that actually. So what happens in the large intestine? Not much. That's it. I mean, other things happen, but not really. So that's pretty much it. Yeah. Okay. Turn the light on.
So is that it for the day?
No. But that's the end of chapter 21. Chapter 22, which you want to sort of get out your notes, we're going to start in literally, guys. There are like 5 things. Okay, so you've got all the stuff on there. There's going to be 4 things on the board and 1 PowerPoint slide. That's the whole chapter. Okay, and a lot of it is actually review from what we did earlier. So that's why we're going to do most of it, because it's nothing that hard at all. It's simply, I'll try to write neatly enough so that you see it. It's simply almost a memorization of a couple things. But we'll review that next time too. But it's so easy that I want to get that done because again, Wednesday is conceptual. Okay, that's when we actually get to something harder than this. This is all nuts and bolts. Okay. You're not good with that pen thing. I'll get it for you.
Thank you.
I like your haircut.
Oh my god.
I'm getting it.
Chocolate.
metabolism, I decided to pick that picture. All right, so let's clear up the board here. Again, this board is the chapter, except for one PowerPoint slide. I can actually leave that up for a second. All right, so this time I ate a breakfast burrito. We just digested and absorbed it like we just saw. Now what? Okay, well, the first thing I want to do is basically talk about sort of what controls your hunger versus being satiated. And there's a lot of things that control it. That's why no one has a perfect diet pill here, because we have basically hormones and the nervous system systemically controlling things. Now, the good news is, and I'll kind of put this up, that literally this whole 10 slides are going to be summarized by 2 columns. What makes us hungry? So hunger, and, well, we're going to say satiated. You know, we're not as hungry. Okay? Now, again, you know, we can eat more. There's all the psychological things that make us want to eat just to eat. But as far as physiologically signaling that we should eat, we're looking at this. With the, in fact, it says there, the hypothalamus is being the center, the hunger center. The hypothalamus, we're going to see, you know, Wednesday as well, very important structure, kind of a combo, an interlink between the nervous system and endocrine, those 2 systems up at top. And so, you know, it's clearly involved in basically the feeding center of the, you know, of the body, of the brain. Now, you know, the first thing is sort of these 2 theories, but they're all true. They all combine together. The glucostatic theory says it's glucose that's controlling hunger or not. And we should be able to figure that out overall. If you have high glucose versus low glucose, which one will make you hungry? High glucose or low glucose?
High.
Low. You like to do that, don't you? Okay? And in fact, while let's put this up here, we're going to revisit this. Blood glucose. But for hunger, it's low blood glucose, because clearly we need to eat then. But if you have a lot of sugar in your blood, it's going to signal. It literally has sugar sensors in the hypothalamus that will will stimulate feelings of satiation, you know, decrease hunger. So high glucose, the opposite. Now, we're going to rewrite this. So we did blood glucose homeostasis at the beginning. And we have this idea that maybe you can get up to, you know, higher than normal, but just higher than normal in general. But if you go too much, we'll just write hypoglycemia. Okay, what organ? Pancreas. And what cell type? Beta islet. So, because this is going to be one of the figures here that we're going to fill in. The pancreas, it's the beta islet, are going to produce what? Insulin. And I'm going to have to, God, that sucks. Hang on a second. I'm going to rewrite it. I just need more room here. So blood glucose, higher than levels. We have pancreas, which are the beta islet, produce insulin. 1, 2, 3, 4, 5. By the end of this part, we're going to list 5 functions of insulin, but not yet. Let's just fill out part of this bottom one now. When we have low blood sugar, we're going to also have the pancreas respond. And it's now the alpha islet, and it produces glucagon. 1, 2, 3, 4, 5. Okay, once we fill that in, that's going to be one of the figures we're going to need to know. Right now, though, it's these hormones. So not only do sugar levels affect the hunger and satiation part of the hypothalamus that have receptors, the hormones do as well. So we should be able to figure out which goes what. When you're hungry, okay, which of those hormones? Well, let's just another one. Let's say insulin. If you have high levels of insulin, is that going to make you hungry or is that going to stimulate satiation? Yeah. They go together. Okay, so it makes sense. If you have low blood sugar and therefore glucagon, it says eat. If you have high blood sugar and therefore insulin, it says you've got enough. Slow down, okay? Physiologically at least, not psychologically maybe. There's a lot more in psychology.
Sorry, just to confirm, if that's hyperglycemia, is that so insulin is just being produced when that happens?
No, the insulin's being produced as soon as we get above that homeostatic range, even before we get to the level of hyperglycemia. Okay? And remember, we always have sort of levels, of it, but it will increase at that. So even the book even calls this sort of the fed state and the fasting state. I mean, it's all relative. But it's just when you get beyond this range that we have is when these things are stimulated. Fat. The other major theory, again, they're all actually combined in reality, is called the lipostatic theory. And the idea of this is fat is our major energy storage. And fat is a huge gland. I've probably mentioned this before. It puts out hundreds of different hormones, and if you have too much fat, they're actually causing a lot of problems. But this was sort of the first hormone discovered, produced by fat, and it's called leptin. And if you've got a lot of fat, it means you don't really want to eat as much. And so here's the idea then. Fat cells produce leptin. I'll put fat cells. And basically that's going to be satiation.
When they're produced?
The idea is when they're secreting leptin, okay, it's going to go to the hypothalamus, and actually I'll have a figure here in a second that will say, don't be hungry, don't eat anymore, satiation. Now what it actually does, and we'll show a better figure, is it inhibits a hunger peptide. And I'm going to show a figure. So, again, we're going to put NPY, which is neuropeptide Y, on this column for hunger. So NPY is a hunger hormone. Hypothalamus makes us hungry. When you have leptin, you inhibit it. Okay, so if you inhibit a hunger hormone, it's satiation. We'll show there one figure on this. See if this flow chart helps do that. But right now it's just understanding the three of those columns. So here they're saying, okay, hey, we're eating. That means overall we're stimulated for hunger. We eat more food. We store more fat. Therefore, more leptin is secreted, and again, this means inhibits a feeding hormone, neuropeptide Y. So it's this sort of feedback loop, which sort of makes sense. It's kind of homeostasis as well, right? So that's those three. Okay, so both of these are involved in kind of the glucostatic theory. This is sort of involved in the lipostatic, but these are basically the columns. Now, is that all that controls hunger? No. There is like this chart and thousands more. This is why it's hard to come up with the perfect diet pill. And the only other ones we need to know are these other ones I listed here. And for the hunger column, when your stomach is empty, you think that should make you hungry? Yes, Mike, it will. And what it secretes is something called ghrelin that goes to the hypothalamus and says, we have an empty stomach, we're hungry, please eat. So in the hunger column, we write ghrelin, okay? And you can also put empty stomach to help us remember. When food enters the small intestine, okay? We went through this process just 20 minutes ago. Food has entered the small intestine. Do you think that should make us eat more or less? If you have a bunch of food entering your small intestine, do you think you want Why would you want to eat more? You want to eat less.
Because your stomach is clearing out.
No, but I mean, this is, it's part that's the digestion, actually, it's not even clearing out, it's sort of storing it. But the idea is that when you're actually having food, it takes time for that to process. We want to slow it down. You've got plenty of food. We actually want to decrease food intake. The small intestine basically makes these two peptides when food enters. We're going to have a chart on that coming up. So the small intestine will make CCK and PYY, sorry, letters, and they basically will signify satiation. So this finishes off our columns, by the way. So we're going to put CCK, which is sort of the main one we're going to see, PYY, okay? This for our two columns. These, again, are made from the small intestine when food enters. And that's the chart that we need to know. Okay? So, hunger versus satiation. And these are pretty straightforward. This is like a note card almost. The only thing I can confuse it with is if I use the words agonist or antagonist. Okay? If I give a leptin antagonist, is that going to make you hungry or satiated? Hungry. If agonist, you stay in the same column. Check. That's number one. Remember I said there's 5 things, so to speak, and we'll probably get through them today, that are really on the board, except for this next slide, that are simply you need to know. So here's the first one. Hunger versus satiation. All these other, oh actually this is going to be the second one, because then I have other pictures of this, but all this is just on there. This is the second thing. This is only on your PowerPoint. Okay, but it's on your PowerPoint. And it's listed so well here, I'm just going to go through this. This actually comes from a different book, but actually this is a great figure. So this is number 2 that you have to know. And what this is going to talk about is, whereas this is systemic control, this is local control. And this is going to make sense from what we just learned. So when food enters the small intestine, okay, you can start to follow this here, it's going to make and secrete 2 things, CCK and secrete. Okay, this is when food enters the small intestine. Now obviously when food enters the small intestine, we want all that stuff we just talked about on the board to go, right? We want the bile to get in and everything to be activated. These hormones are going to help control that. So let's follow what's happening here when we go through this. So first of all, we have this idea of what just happened with the gastric juices before, and we have this idea that chyme is entering the first part of the small intestine, the duodenum. And that causes the release of these 2, secretins and CCK. Now, the next 3 bullet points say what they do. They both inhibit upstream process, right? If we have food entering, we want to take time. We don't want more of the stomach to be active, so we want to decrease the activity. So it decreases the activity of all the chief cells, parietal cells, etc., so they're not secreting as much. We want to slow down. And so it's local control now. These hormones, CCK and secretin, will do that. That's basically that. Now, CCK will get the bile to come in and the zymogens. Secretin gets the member of pancreas released also bicarbonate. Secretin will basically tell the pancreas to secrete bicarbonate. And that's that. So it's a very good figure, and so I think it lists things very well, and that's what you see. Again, remember it's not an essay at all. That's what you sort of have to know. Sort of the action of these 2 hormones, CCK and secretin, of what they're doing here. Basically, we're saying food is entered, it's time to slow down the upstream process, and everything we just talked about in the previous chapter, we want to get these secretions in to do their job. And these 2 hormones will do it. Local control. You know, a lot of this course, as we get to the end, revisits concepts we did at the beginning. We're going to be doing a lot of homeostasis, negative feedback, next time, and also this idea that when we look at control in the body for homeostasis, we look at systemic control with the nervous system and endocrine system, and we look at local controls here. Okay, we saw that with blood pressure, etc. too. You're seeing that again in the digestion metabolism. That's 2. Okay? Remember I said 5 things, so to speak? This one is, I'm not writing on the board because it's sort of there, but that's that one. 3. 3 and 4, I'm going to be using these. And that's going to be the bulk of it. And then we just talk about diabetes, and we're done with both chapters. So both of these chapters, again, are just almost straightforward. You almost wish the course were this way. Now I'd be bored. All these slides are just these, you know. So we look at this for a second, and then we draw this up here. This is sort of the overall metabolism. The only reason I want to show this is we're going to be putting these terms up, basically, on this. And so we're going to be talking about lipogenesis and all that. But I like to show this because, again, it relates the idea that our central metabolite, glucose, and why the brain needs glucose. And so, we get glucose, we'll use it for energy for the brain and some other tissues. We'll store the excess as glycogen. If we have too much, guess what? We're going to store it as fat. So, sugar can be made into fat. Overall proteins, the absorbable units are amino acids. We don't want to use them for energy. We want them to make proteins under normal conditions. So it does that. Fat, of course, is going to be used for energy and storage. Okay. So with that said, let's look at this now. We're gonna have to do part of it over because I need to clear up some of this. We have carbs, proteins, and lipids, triglycerides. We actually, everything we're writing up, we actually did in chapter 4. And so what we're going to do is write names. And when we're doing these, now we have to know that not only are we breaking things down, actually, I'm going to want to actually make this glycogen. So we're going to talk about the main thing for us, because we don't have starch in our body. We're going to store it in glycogen. We're going to, I guess we'll break things down, break it down into glucose, but we're also going to store it as glycogen. Now we'll break down glucose to pyruvate, and by a series of steps, CO2 for energy. We are not plants, so we can't make CO2 into glucose. Sorry, but they do it for us. Proteins, we're going to break down into amino acids, but we're also going to synthesize proteins from amino acids. This is our metabolism. Lipids, we're going to basically break them down into fatty acids and glycerol, and of course we're going to put those together to make fats. In general, when we are breaking things down, these are catabolic reactions, and we're going to see that they often end in the word lysis. Again, all this we actually did, but now we have more perspective. When we're making these things, these are called, and we're going to use the word genesis. Okay? So, let's name them. Well, let's start over here. Okay, when we're going to break down lipids into free fatty acids and glycerol, we're breaking down, it's going to be lipolysis. I don't care, lipolysis, whatever you like. It's simply lipo with the lysis. When we're making it, lipogenesis. Now proteins, we break them down, proteolysis, proteolysis. Usually we just say protein synthesis value. And I'll just say that. You know, you might see proteogenesis somewhere, but you really won't see it too much. Protein synthesis. Proteolysis, protein synthesis. Okay, these two, glycogen, glucose. The key is, don't start putting glycolysis here, because that's coming up next, is that you're breaking down glycogen or you're making glycogen. And you just use those suffixes. So if we're breaking down glycogen, it is simply glycogenolysis. Okay? Glycogen with an O lysis. If we're making it, glycogenesis. Okay, to finish a couple, I have like 3 more little things there, and then we'll be kind of done with this third figure, so to speak, of what you need to know. All this is sort of nothing. All these words we are actually fitting in here. That's why I mean, a lot of this slide or a lot of this PowerPoint is just made into my board here. All I want to show with this, because we don't have to memorize this for the class, is when we talk about this whole process of converting glucose to CO2, getting energy. Most of you have had this in general bio and other classes. It's several processes here. The first process is by a number of steps is to convert glucose into pyruvate. This doesn't require energy. It doesn't require oxygen. It makes low energy. This is called glycolysis. So this sort of first stage here, converting glucose to pyruvate, is glycolysis. Now the rest of the way, it goes through the citric acid cycle, electron transport chain. That's where we need oxygen. We're not going to know, there's 20 steps or so. All we have to know from getting over here is we're using oxygen. And so this one is just aerobic respiration. One more term, this figure is done. And I'll get out of the way. So it's not shown on that slide, and we had this in the first section too. Fatty acids are these long carbons. We can't feed that in. We do feed fats into the citric acid cycle. We can't feed that whole long chain. What we're going to do is break it down into 2 carbon units and feed that in. And breaking it down into 2 carbon units is called beta oxidation. That's breakdown of fatty acids. Glycerol on here so we see. It's the breakdown of fatty acids into 2 carbon units. That's our last term, beta oxidation. And this is the third of the 5 things, is knowing these names for the synthesis and breakdown of carbs, proteins, and fats. Which, again, we actually had that earlier on, but now we're going to, not only are we going to put it in perspective, but we're going to use that for our number 4. Which is probably the last thing I'm going to do today. The 5th thing is just talk about diabetes. We'll put that up next time. Probably. Any questions about this? Okay, for this. Well, so we're going to go back again. We're going to show you 5 basic functions of what insulin and glucagon do. At the beginning of the course, we did a couple. Now we're going to combine that board with this board, and that's sort of the key with this. So, and again, the book kind of puts this as the fed state. Again, all these figures are unnecessary because I'm simplifying it. I'm going to come back in a second to that. We're going to kind of do something like this, but I'm not using, you want to look at my terms and what I'm going to describe, and I'm putting 5. But we're talking about sort of this idea of what's going to be happening when we're in the fed state or when blood glucose levels are gone up, and what's happening when when blood glucose levels have gone down. How it's affecting all of this. And we already know it's going to be mediated, these effects are going to be mediated by these hormones, insulin and glucagon. For insulin, I almost want a third board, but we can kind of put it over here. Overall, if you take here's our cell, here's glucose, which is in the blood. Okay? We want to get glucose in the cells. Can glucose just diffuse through the cell?
No.
It needs a membrane transporter. Now, these membrane transporters, which are these glue transporters, most cells, you can't get glucose in. Those transporters are not active. You need insulin to bind to its receptor and activate these. You're not going to have to memorize all these steps. But this is what insulin is doing for the first sort of step. We talked about increased glucose entry in the cells. So it's binding to its receptor and therefore allowing glucose to enter through its. And so I don't really care about sort of the details, but and that's what we put originally. Okay, what does insulin do? It's going to up arrow increase glucose entry in the cells. So the details are important, but I do want you to realize, because when we get to diabetes, the 2 slides on it, which maybe we'll just do today, we do care about this idea of insulin needs to bind to its receptor to activate the ability to get glucose in. Okay? Again, the steps in the middle are not important for us, but you do have to realize for these glucose transporters to work in most cells, you need insulin to bind to its receptor to activate. Okay? That's the key. Now, once glucose gets into the cell, we actually wanted, we got it into the cell to do this, to do all this process so we can make energy. Okay? And so this involves all these enzymes involved in this glucose breakdown. Insulin actually promotes that. So insulin doesn't just get it in. Okay, and I'll kind of go back to the last figure. It doesn't just increase the entry into the cells, it increases glucose breakdown. So that we can actually extract the energy. So, increases glucose, this is insulin itself doing this, increases glucose breakdown for energy. That you can convert, you can extract the energy from glucose by breaking it down. We call that, by the way, if you look up at this top one, glucose oxidation. So this whole process of breaking down glucose so you can get energy, that's sort of why you can look at that term, it's called glucose oxidation. And it does promote that directly. So it increases glucose entry into cells and increases its breakdown. Okay. 3, 4, and 5. Okay, we've got a lot of energy, right? We just ate, we've got all this energy. I think I'm in an anabolic type of mood. Isn't this when you would make things? Okay, if you have a lot of energy, a lot of glucose in the blood, wouldn't this be the time that you have the sort of the luxury to store it as glycogen, to make protein, to store things as fat? Insulin promotes all of those things. This is why insulin isn't just doing what we thought. I mean, most people knew it was that, but it literally, both these hormones, affect our entire metabolism. That's why I like having this up here, because we can literally put that, okay? For 3, 4, and 5, insulin is going to increase protein synthesis. We're just putting all those over there now. Okay, 4. it's going to increase the making of glycogen, which of course we call glycogenesis. 5, it's going to increase the production of fat, which we call lipogenesis. So 3, 4, and 5 is this, and this. Okay? The insulin, that's why, you know, too much insulin can make you fat. Okay? Not only is too many calories going to do it, but the idea is insulin itself promotes the formation of fat. Okay? So insulin, you know, again, which again, most people just wouldn't realize this, you do it, affects your entire metabolism, not just kind of what we know gets glucose into cells. It affects the breakdown of it so we can get energy of it in those cells, and then the rest of our metabolism, it actually helps promote all these anabolic reactions. And we're done with insulin. Glucagon means we don't have enough sugar for the brain. We better do some things to correct that. The main site of action, so this glucagon will act on mainly the liver. And mainly from the liver, this is, you know, glucagon's action on the liver are going to do these 5 things. And this will be figure 4 out of 5. So, what are they? Well, we need glucose, okay, for the brain. Where do we store glucose?
Liver.
Exactly. And so it's going to promote the breakdown of glycogen into glucose, which we call glycogenolysis. We break down glycogen into glucose, and that feeds glucose into the blood to feed the brain. Okay, everyone, that makes sense. Okay, so sugar, other tissues can use these other food sources. The brain needs glucose. Seems like what we want to do now is break down we want to break down proteins, break down fats, so the other cells can use that for energy to save glucose for the brain. That's what this enzyme, or glucagon enzyme, promotes as well. It's going to help break down proteins, which we call proteolysis, and break down fats, which we call lipolysis. Okay? That's why those terms are there. So it's basically lysis, 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 and we're going to put these last 2 here. We're going to break down glycogen into glucose, feed the brain. We're going to break down proteins and lipids so that the other tissues can use that for energy to save glucose for the brain. 4 and 5, 2 special things. And that's where we'll stop for today. There's only going to be 2 slides on diabetes, which you can almost look at, but we'll do. That's the chapter. So 2 sort of emergency things, which I'm going to describe, that the liver basically will promote. Especially, again, these are sort of, more sort of desperate measures to make sure we stay alive. And those 2 are, I'm going to write him here, and then we'll describe them. Gluconeogenesis and ketogenesis. And these are the following. So gluconeogenesis this is glucagon on the liver is going to promote this. We're going to basically convert amino acids, let me put it this way, convert non-carbohydrates which are mainly going to be these amino acids and glycerol, sort of the breakdown products we already made. The liver can make those convert them into glucose to feed the brain. That's why it's called gluconeogenesis, okay, like a new. So some of these products, not only are they being used by the other tissues, we can also use them as building blocks to make more glucose. And that's gluconeogenesis. The last term at the end.
So it's kind of like a backup plan.
Say again?
It's kind of like a backup plan.
Well, it's I don't even call it a backup plan. It's an extra plan that's part of it. It's in fact that's a good question. Let me write this and I'll actually sort of address that, because it's actually a good point that a lot of people ask. So let's put this other one and we'll put them together on that concept. Ketogenesis. We're going to make things called ketone bodies, or ketones, from fatty acids, which are the other breakdown. And these, again, these are sort of what I'll put as emergency fuel for the brain. They don't like to use fats, but we want to keep alive. The problem with that, though, is these are acidic. And so you're prone to ketoacidosis. Which is, of course, what type of acidosis? Metabolic. What cell types respond to the kidney? No, we don't. Ketoacidosis. So these are sort of, again, more of an emergency fuel for the brain. They're fatty acids which can circulate and the brain can use. Now, and that's why we have it here. The sort of what I hope is potentially answering your question too is that we're obviously using everything here. And so some people are like, well, yeah, do you get rid of all the glycogen and then do gluconeogenesis and then do ketogenesis? What actually happens is that, as soon as you get below sort of our homeostatic range, all three processes, all these are activated. But you're preferentially activating this versus this versus this. As you start running out of glycogen, you rely more upon the gluconeogenesis and ketogenesis. And again, the more you're starved or whatever's happening, the more these will start taking over. So they all actually are happening, right away, but it's just that preferentially this is happening. But we are being very efficient. We're breaking these things down so that other, the proteins and lipids, so that other cells can use it, and we're using some of those byproducts to also, help us survive as well. I'd say that's pretty efficient. Any questions? Okay, so again, I'm going to have a student. We actually have to write this number down. Again, you can leave without reviewing me if you want. It's up to you. But anybody that wants to actually review the class, she'll hand them out, spend a few minutes on it, and she'll collect it. I'll go over after that to the lab, and people can look at tests or talk to me about anything else. I just have to write down your CRN when it asks. Just write down the Monday one, 56948.
Can we get one?